the elders past, present and emerging of the Aboriginal country our university is situated on. And that includes, of course, the Darug, the Gadigal, Darawal and Gundungara peoples. And I'd particularly like to acknowledge the Burramatagal people of the Darug nation, on whose country ICS is physically located. The country of none of these groups has ever been ceded, always was, always will be Aboriginal land. Now, I think you probably already know that Eban is an anthropologist. He specializes in multi-species ethnography. And in today's lecture, he'll be directing his attention to COVID-19, bringing his particular expertise and intellectual orientation to bear on what it is and what it does. I think, I hope I haven't misrepresented what you're going to do. Among Eban's many publications are 3 books, which I'll mention. The Mutant Project, his latest book, published last year, his Emergent Ecologies, which came out in 2015, and his book, Freedom in Entangled Worlds, of 2012. Eban also writes on human rights issues and environmental justice, and a number of other issues, I think, for newspapers, which include The Guardian, The Atlantic, and Wired magazine. So, you know, he very much has a presence inside and outside academia, and he has a personal website, which I would recommend that you have a look at. With that, over to you, Eban.
Well, thank you very much for that kind introduction and for the invitation to speak today. And I'm going to do sort of a 2-part talk. And in the first part, I'm going to introduce the idea of multi-species ethnography and just kind of give you a sense of where it came from and where it went along the way. And towards the latter half of the talk, I'm going to use this approach to get inside of the current moment, the moment that we've all been living through over the last year, the pandemic that has emerged with the coronavirus. So as Dennis said, I'm an anthropologist, and in many ways, work about animals is nothing new. There were a series of very classic studies, including one by Lewis Henry Morgan, someone who was in some circles credited with founding the discipline of anthropology. Morgan wrote a whole book about the engineering culture of beavers. And in some ways, you can read this book as an animal rights treatise. It was written at a time when beavers were undergoing massive declines as a result of the fur trade. And he was interested in exploring, again, the culture of organisms that he saw as clever animal mutes. Later on in the 20th century, animals became of prime symbolic importance to anthropologists. Levi-Strauss famously talked about how animals are good to think with. Marvin Harris, a materialist, countered that they're actually just good to eat. And more recently, Donna Haraway, with her book, When Species Meet, insisted that we reckon with animals as agents that are good to live with in common worlds. So the Multi-Species Project in anthropology very much draws inspiration from Haraway, but also broader thinking in the discipline of science and technology studies. So this is a classic study I'm referencing here by Bruno Latour, where he was thinking about non-humans as agents. And Latour, throughout his career, he's dabbled in the realms of he's written books about microbes and other other organisms, but is most known for his work on non-human actants that produce inscriptions about the world and let us build these networks. So, you know, we're in part in dialogue with Latour as well. So the special issue of cultural anthropology, where we introduced this term, multi-species ethnography, Stefan Helmreich and I were interested in the ways that we might understand how other organisms' lives are shaped by political, economic, and cultural forces, but also how they shape what it means to be human. We're really looking at, you know, what Ian Hacking would call these looping effects, where actions and intra-actions across species domains generate emergence, generate unexpected dynamics in the worlds that we live in. And, you know, it's a fact of life, right? We are constantly interacting with other species, whether those are the microbes in our gut or whether those are the kinds of organisms that we eat and are connected to through these assemblages, these commodity chains that bring sustenance to our modes of being. And rather than just celebrate these basic facts of life, it's really a question of justice at the core of multi-species ethnography, this question of who benefits when species meet. And again, this very much comes out of Donna Haraway's work. Another scholar who has been very influential in my own thinking is Susan Lee Starr, who posed a similar question way back in the 90s. We've also, in some ways, tried to depart from Bruno Latour. And again, dialoguing with Susan Lee Starr here, we've tried to get away from this idiom of the non-human. So in many ways, the idiom of the non-human reinforces human exceptionalism. So in Starr's words, non-human is like non-white. It implies the lack of something. And part of what our project has been about is specificity. We're writing at a moment when you know, nature and culture were seen as separate domains, when the science wars of the 1990s made biologists incredibly suspicious of cultural anthropologists who were intruding into their domains, but also made anthropologists sort of wary to think with the biological. So along with Haraway and many others, scholars in the multi-species field have tried to, again, think about the specificity of other organisms. So, you know, one concern was anthropomorphism. We don't want to project, you know, what it's like to be human on other kinds of life. But at the same time, we don't want mechanomorphism. We don't want to assume that others are like machines, you know, a mode of thinking that you can trace back to Descartes and other thinkers. So again, it's about the multi-species idiom is about attending to the specificity of agency and interaction, to think about these dynamics that emerge when species meet. So in this initial collection of essays that we assembled for cultural anthropology, one notable essay really explored some of these key ideas about interaction amongst different domains. And Celia Lowe's essay was looking at how a single base pair mutation to a virus, in this case H5N1 avian influenza, produced dramatic shifts in human migration patterns and the ways that we interact with poultry. She was particularly interested in Indonesia and Southeast Asia, but also as it reverberated in realms of discourse. Celia's article was about viral clouds, about the uncertain ontologies and epistemologies that emerged at this earlier moment when a pandemic seemed to loom on the horizon, something that never quite actualized into you know, the terrifying dreams that people had, but you know, this imagined disruptive event that would radically reorder life on Earth. So she traces very carefully how political and economic institutions, how the velocity and intensity of capital accumulation basically created the conditions of emergence for this particular strain of influenza, and then looks at how that particular strain reverberated out and amplified itself within modern assemblages. That original collection with cultural anthropology also considered modes of interacting with other forms of life that really tried to destabilize human modes of perception and action. Again, trying to get past this binary between anthropomorphism and mechanomorphism to think about other ways that we might interact with creatures on their own terms. So Eva Hayward offered us a piece about the fingery eyes of cup corals, organisms that don't have eyes, that don't have even mouths and anuses in the same way that we do. They have a single orifice that you can see here where everything kind of goes in and then back out again. But more importantly, she was trying to get us to think about the haptic way that we can interact with others, about how we can touch, taste, feel, and come to know through what she termed fingery eyes. So modeling her own mode of interacting with organisms after the organisms she was studying. She did the care work of interacting with these cup corals in the laboratory, but also attended to the ways that cross-species encounters can produce sensations and how we might sort of depart from those sensations to rewrite the species interface. So we also started to explore questions with artworks. And, again, we were trying to displace the sort of the antagonistic dynamics of the science wars where you had biologists and anthropologists sort of at war with each other over nature and nurture. We found the terms of that debate to just shut down interesting kinds of conversation. And we found this idea of George Marcus's, of para-ethnography, to be really generative and productive as we entered into collaborations with biologists and artists, other sorts of experts with shared, discovered, and negotiated critical sensibilities, to quote from Marcus's original piece. So if you look at the word para-ethnography, you can think with paralegal or paramedic, to think about the kinds of work that is associated with established credentialed lawyers or established credentialed doctors. There's other kinds of experts that aid in the triage work in a legal office or in an ambulance before the patient arrives at the hospital. So we worked with artists kind of on those terms, both entering into their domain and inviting them into ours, thinking about how artists might help us do the work of ethnography to identify how power is working in distributed biopolitical assemblages, but also how we might come to know through making, how we might start collaborating with artists, thinking with Jim Clifford about the moment when anthropologists started writing culture. Anthropologists at the Multispecies Salon began making culture by hosting performative events, by curating exhibits, and even crafting original artworks. And the particular strand of art that we engaged with was the tradition of tactical media. And the idea of tactics, you can trace back through Garcia and Loving to Michel Desserteau, who talks about the distinction between tactics and strategies. And in this particular arena, we were interested in ways that we could do de-skilling, how anthropologists could unlearn some of the assumptions that they had, how artists could also unlearn some of the assumptions, but also reskilling, the sorts of skills that we might share with one another in this DIY space. And in thinking with Beatriz de Costa and others who've talked about tactical biopolitics, the project of the Salon came to be exposing how biopower is functioning, exposing how these dominant assemblages are configured, but also not just doing the work of critique, but making concrete onto epistemological interventions by re-articulating assemblages and putting them on display for critique and inspection. One particular artwork that I still find very generative, and it kind of goes back to the viral theme again, is Caitlin Berggren's Life Cycle of a Common Weed. Caitlin was infected with hepatitis C when she was a young girl, and now she does performance art pieces where she uses her blood, something that would be harmful to other humans, as a way of giving nutrition to plants. She remembers a moment as a kid walking into a hardware store and seeing a bag of blood that was about the same size and weight as her own body, and just imagining herself rendered into biological substance that would give to other organisms. So in that hardware store, it was basically dried blood, from a slaughterhouse being sold to organic gardeners. And with this piece, Life Cycle of a Common Weed, Caitlin is nurturing a kind of plant, a dandelion, that others have rendered killable. Dandelions are commonly regarded as weeds. They're popping up all over the place. But it turns out they are a source of medicine for people with liver problems like hepatitis C. So she takes dandelion root as a medicine, and also feeds the plants with her own blood, creating what she calls a microbiopolitical network, a circulation of matter and meaning that basically is outside dominant regimes of regulating biological matter and life. She describes it as a pathetic political gesture in giving back in this way to a plant. It's really asymmetrical in a sense to give a little bit of blood to a plant when you're uprooting it using those roots for medicine, killing the plant. But she wants to, make that gesture to attend to the ways that all kinds of life in this world is rendered killable. Another contribution we had in the Multispecies Salon edited collection that was published with Duke was from Karen Barad, who talked about invertebrate visions. And she used the brittle star, a relative of the common starfish, but one that is distinct. And there's a number of really peculiar and interesting things about the brittle star. Her essay centered on the biomimetic initiatives that some technologists have developed. So brittle stars don't have eyes, they are eyes. The exoskeleton of the brittle star can sense light and respond to it, not by routing signals through any kind of centralized brain or ganglia, but by becoming with the world, interacting in dynamic real time. As shadows appear, limbs retract. But there's also other remarkable features. If a limb breaks off, the whole organism can regenerate. So she was using the brittle star to illustrate one of her abstract ideas that she's developed in the realm of particle physics, to think about these kinds of interactions that organisms can have with their environment. And it's I think an idea that can extend out from this particular organism, this particular interaction, to account for other kinds of multi-species dynamics. So Barad accounts for ways that interactions are a marking of the relational nature of the world and its becoming. She's describing agential realism, where agents and subjects only emerge in this interaction. It's a concept that's different from interaction. It's not like two billiard balls bouncing off, remaining stable as they take on a new vector or trajectory, but it's a way of thinking about the transformative nature of encounter, about how when two things encounter one another in the world, they're changed and go on to create reverberating changes in the world after that encounter. So as the field of multi-species studies started to get established, it started to attract some criticism. Among others, Tim Ingold took aim and suggested that we abandon these approaches. He insisted that the notion of species is an anthropocentric imposition. Ingold wrote, "Only in the purview of a universal humanity does a world of living things appear as a catalog of biodiversity, a plurality of species." And as Ingold wrote that, I was doing some ethnographic work actually in Sydney in the Royal Botanic Gardens with people who were actively doing the difficult work of recognizing and differentiating amongst different kinds of life. So one of the key people I was working with, Dale Dixon, is a taxonomist mostly of fig trees. So figs are an iconic feature of the Sydney landscape. The botanical investigations into figs go back many decades, even hundreds of years here in Australia. And conventionally, what people would do in this field of botany is look at the morphology of the leaves, of the flowers, and yeah, maybe impose an idea of what a species that groups together similar organisms might be. But the species concept has really changed. You might think with John Dupre, who's a philosopher of biology, who calls for a promiscuous realism in accounting for species, basically insisting that in each given branch of the taxonomic tree, the idea of species is different. It's tailored by these different kinds of biological experts. So Dale Dixon, who is a practitioner, very much has a theory of species that fits with this promiscuous realism of John Dupre. He insists it's not a universal idea, but you must develop an idea that fits for the particular group or clade of organisms you're interested in. And what Dale really does is scrutinize multi-species interactions in determining what constitutes a species. So if you go to the Royal Botanic Gardens in Sydney, you'll see this iconic fig tree, Ficus macrophylla, subspecies macrophylla, commonly known as the Moreton Bay fig. Right nearby, you'll see this banyan tree collected from Lord Howe Island, which is in the middle of the ocean between New Zealand and Australia. Basically this sprawling form, instead of having a single trunk, it has a multiplicity of what you might term, following Deleuze, aerial rhizomes. There's no clear points and positions. It's sort of morphing and growing. But Dale did the very careful work to determine that these are the same species. He followed the flight of wasps that pollinate the fig flowers. So this is a picture of those wasps taken from one of those trees. And you see in the middle, one of the wasps is burrowing inside. So if you've ever opened up a fig fruit, you'll see a bunch of little seeds. Each one of those is a flower. And you might discover either a wasp or a wasp larva inside. That's how figs make future generations. So he basically found that these wasps were taking pollen from one of these fig trees in the Royal Botanic Gardens, the tree with a distinct trunk, to the banyan. The pollen was cross-pollinating, as it were. And digging even deeper into this story of multi-species recognition, differentiation, and categorization, and basically, these wasps will only go to some fig trees and not others. There's a diversity of fig trees in this garden, but this particular wasp species only goes between, these two types or kinds. But digging even deeper into this multi-species story, it turns out that there are bacteria inside of the fig wasps that are shaping the species of the insects, of the wasps. So there's a bacteria called Wolbachia that basically is only going to make more of future generations of insects when there's a compatibility. So if you have two different Wolbachia strains infecting different kinds of wasps, those are going to speciate and produce different wasp species. If you're going to have one strain of Wolbachia that's in a fig wasp male, but not in a female, that will produce a dead end. Basically, the story that Dale tells is that these microorganisms are shaping the boundaries of the insect populations, of the wasps. The microorganisms, the bacteria, are engaged in that work of recognition, recognizing themselves, singing species into existence in future generations. At the same time, the wasps are singing the trees into existence, calling forth these future organisms. So I came to understand species as entangled agents, as phenomena that emerge in these intergenerational dances among entangled agents, where they're torquing and twisting one another in ongoing loops of multi-species interactions. Here I'm thinking with Barad again, but I'm also thinking with Ian Hacking, who describes the looping effects of categorization and making up different kinds of people. You find that the categories themselves change the people. So these practices of multi-species and interspecies recognition were also shaping the kinds of creatures across these taxonomic lines. And, in thinking with Dale Dixon, I found that what he was trying to do is recognize these different agential beings and look at how they were doing their work. And, panning out from this particular case study, I became interested in how species emerge, how they disrupt human practices, political and economic systems, as well as ecological communities. You can see how new kinds of critters are generating order-forming assemblages, as well as order-destroying disasters. So that brings me to the pandemic and the emergence of SARS-CoV-2, the virus that is behind the COVID-19 pandemic. And for starters, I'd like to make a key distinction. COVID-19, the disease, is a separate thing from the virus, SARS-CoV-2. So you can get a positive SARS-CoV-2 test, but never come down with COVID-19, the disease. That's a really important distinction that a lot of the virologists who I'm interviewing these days want to make. Early on in the pandemic, I published this story about the emergence of COVID-19, basically taking a critical look at some of the evidence linking back coronaviruses to the novel coronavirus. Basically, when you push hard at that dominant outbreak narratives related to so-called wet markets in China, as well as conspiracy theories about the Wuhan Institute of Virology, they fall apart. I tell a messy story of emergence, and a story that has many gaps in it still a year later. The early reports that we saw about bats and pangolins are reporting on viruses that are distant relatives of SARS-CoV-2. If you do the gene sequence and bioinformatics work, you find that the most recent common ancestor between the bat virus reported in this paper and SARS-CoV-2 was 1981. So there's been a lot of dynamics and transformation and change that have taken place in the last 40 years. So a few weeks ago, when I guess now about a month ago, when the World Health Organization visited Wuhan, they found many different strains of SARS-CoV-2 that were out there circulating before the pandemic was detected. Now we know that relatives of SARS-CoV-2 have been circulating around multiple animal species for decades. It's unlikely that we'll be able to pinpoint a single moment of emergence, but I'm interested in how all these different forces, animal, technological, capital, all these different things have contributed to this moment of viral emergence, multiplication, and the subsequent dynamics of the pandemic. So when the WHO team visited, they visited the so-called wet market. They interrogated evidence related to serology. Antibodies to the virus were found in patients, before it was formally detected. But importantly, again, there were all these additional strains that were found. So there was at least 13 additional strains besides the original reference sample that have now been sequenced. And it's still early days to figure out, is this something that emerged elsewhere in China? Is this something that emerged, in Southeast Asia, or possibly even Europe? The report from the WHO details a number of studies that have some evidence of SARS-CoV-2 in places where it wasn't supposed to be before the official outbreak in January 2020. So currently I'm involved in a project to visualize the virus in collaboration with Sriya Chatterjee, and we're forming an interdisciplinary team of artists, curators, and other scholars. We're collecting images of ground glass opacities in lungs, neutrophil extracellular traps or nets. These are basically exploding cells that create blood clots throughout the body and result in some of the pathological dimensions of these infections. We're also collecting electron micrographs of viral particles. And basically what we're trying to do is think about how viral agency manifests itself on multiple scales, on the scale of molecules, cells, and tissues, before we pan out and think about how it interfaces with other assemblages at other scales. So I think we can understand the agency of SARS-CoV-2 with help from classic science studies texts. Bruno Latour introduced the idiom of co-production in 1979 to think about how scientific apparatuses produce inscriptions, transforming pieces of matter into written documents. Later, Sheila Jasanoff elaborated the idea of co-production to suggest the ways that we know and represent the world, both nature and society, are inseparable from the ways which we choose to live. Much of the earlier work that I've talked about today related to multi-species ethnography, again, has tried to destabilize ideas of agency in more than human assemblages. Together with Stefan Helmreich, I'd helped introduce a new frame for thinking about how animals, plants, fungi, and microbes shape and are shaped by political and economic forces. So if we bring that back to the study of SARS-CoV-2, we can think about how viral agency is co-produced in concert with molecular assemblages inside host cells. In thinking about SARS-CoV-2 as an agential co-production, I think it's an opportunity to twist inherited images and ideas. For starters, I think we can turn Sheila Jasanoff's ideas upside down and inside out to suggest that viruses contain representations of the world. Viruses are inseparable from the ways in which they interface with living systems. So many of you are probably familiar with the ACE2 receptor and the spike protein that binds with the receptor. Basically, this is the port of entry for the virus to get inside of the cell. You're probably less familiar with the dynamics that take place once inside of the host. Viral replication takes place in concert with organelles, enzymes, and metabolic products. In thinking about how viral agency emerges through molecular interactions, it is important to think of a virus not as a static particle, like the electron micrograph that I showed a few moments ago, but as a living process. It's important to sidestep foundational distinctions between life and non-life. I follow John Dupre and Stefan Gudinger, who argue that viruses should be seen as processes rather than things or substances. Building on the insights of Karis Thompson, we could also think about viral replication and assembly as a process of ontological choreography. Agency is distributed through processes that are acting in concert. None of these molecules choreographs the whole show. The viral subject and agent emerges as a co-construction and a co-production in a dynamic molecular dance. So if we go on to look at the specifics of SARS-CoV-2, and that image that I just showed you was not SARS-CoV-2, that animation was a flu virus infecting a cell, but it's a similar sort of process of interfacing with these cellular dynamics. But this is a shot of a key part of the SARS-CoV-2, what's called the proteome. So this is kind of what the virus looks like after it kind of unfolds, unravels, and starts to interface with the cell. So each viral species and family has an idiosyncratic molecular process that emerges within the host cell once the genetic information is translated into this lively assemblage of proteins. And this is kind of part of the key bit of how SARS-CoV-2 works. Part of what has been well described in SARS-CoV-2 is a suite of 16 genes that make this molecule, replicase transcriptase. This is the enzyme that makes new copies of viral RNA in the host nucleus and other structural proteins of the virus that are assembled in other parts of the cell. But thinking across scale and thinking about agency and vulnerability, I think it's important to quickly pan out and gesture to how viral strains become entangled in ecosystem processes, cycling through bodies of humans and multiple animal species. If we're trying to think about the force of viral agency at multiple levels, I think it's important to build on a deep archive of scholarship dating back to Karl Marx that explores non-human agency through hybrid articulations of capital, consciousness, and machines. I'm interested in ways that SARS-CoV-2 was amplified by processes of capital accumulation, food production, global acceleration. As I make these jumps across scales, I'm collaborating with an international team, again, of artists, ethnographers, critical race theorists, virologists, and historians to approach questions related to vulnerability. Rather than just focus on contagion, we are considering configurations. We know that vulnerability to infection depends on the state of a body before a virus arrives. Bodies are shaped by political and economic forces that leave durable molecular signatures. Asymmetries of vulnerability and responsibility have become apparent in the pandemic as Black and brown bodies have become casualties of what Anthony Ryan Hatch calls a necropolitical war. My team of collaborators and I are asking, how do cells, bodies, communities, technological assemblages become vulnerable to disruption by viral agents? How are viral processes transforming the human condition and broader multi-species worlds? Thank you. So I'm happy to take questions. If I was just basically talking to an imagined future YouTube audience, but I'm happy to take questions if anyone wants to have a dialogue.
Justin, we can't hear you. We still can't hear you. I'm having video problems and Justin seems to be having microphone issues. He's going to type, okay.
Okay, so just reading the question here. Great talk. I was wondering whether multi-species ethnography connects up with work that is being done in plant physiology and microbiology regarding consciousness and subjective experience in plants and bacteria. So yeah, I mean, there's been conversations ongoing, and I think one early point of reference really predates the multi-species conversation. Going back to Jakob van Uxtel, a thinker who is associated with the idea of Umwelt or ringed world, if you translate it literally from the German. So van Uxtel was taken up by many contemporary philosophers, Heidegger, more recently Peter Sloterdijk, and in the readings of those philosophers, the Umwelt idea illustrates the boundedness of these phenomenological worlds. So Heidegger famously said, or infamously said, stone is worldless, the animal is poor in world, the human is world forming. Peter Sloterdijk offered his variation of that thesis. Sloterdijk, who styles himself as the second coming of Heidegger, very modest guy, he talks about the human as an ontological amphibian and then insists that other organisms are trapped in these ontological cages. I think if you go back to the van Uxtel and kind of ignore Sloterdijk and Heidegger, you see a very nuanced account of the boundedness of these phenomenological worlds. And I think really good resources that contemporary ethnographers can use to think about the specificity of agency and action in shared worlds. I think it's important to think about the gaps in sort of sociality and subjective experience as much as the overlaps. Anyone who has a dog or a cat or other kinds of creatures in their life, knows very clearly you have a social relationship. I think there's naive realist assumptions that many people often make, that the interior state of a dog is analogous to some feelings that you might be feeling. Some dog expressions might seem to say shame, but if you carefully attend to canid worlds and canid sociality and hierarchy, an emotion that we might read as shame, or a facial gesture of a dog that we might read as shame, might mean something totally different. So I think, yeah, there is a lot of allied work being done, here in Australia, out in Perth. There's phenomenal thinkers about plant subjectivity, people showing that plants can remember being dropped and will, close their leaves or open them. There's, similar work being done related to microbes, and also work related to how human experiences of consciousness might be mediated by microbes. Here I'd point to the Mind the Gut project of the Medical Museum in Copenhagen. So there's a lot of people who are trying to decenter our ideas about human consciousness, and to sort of question any sort of thesis of human exceptionalism as it's embedded in the Heidegger or the Sloterdijk, to think about shared modes of apprehending and responding to the world, but also when you push it too far, I think things break down. So kind of thinking with the virus for a second, in this talk, I was trying to insist that we engage with the virus not as a static particle. It'd be like thinking about a plant, a tree, as a seed. Like, seeds aren't that interesting. I mean, they can endure, great physiological, climactic chemical stress. Like, seeds are really durable, and viral particles are similar or analogous in their ability to sort of exist in environments that are otherwise hostile. But once inside a permissive cell, the virus comes alive. And but I think there's something different between virality, like viral modes of life, and the sort of modes of apprehending a world that a human might have, or a dog might have, or a plant might have. So it's coming back to this idea of specificity, thinking about specificity of agency, specificity of conscious and subjective experience, and really treating biologists as useful allies, critical friends, in the idiom of Jenny Reardon. So if there were moments in the past 30 years when biology and anthropology were kind of at odds with one another, we're now at a moment where, we're seeing the strategic borrowing or poaching, to invoke Desserteau, where we might go into the domain of biology and, maybe we're not going to accept all of the premises about, evolutionary fitness, or, the teleology of anagenetic directed evolution. But, we can still poach and treat seriously the knowledge, the specificity that particular fields of biology have developed to think with their particular organisms. So that idea of critical friendship leaves open the possibility of kind of an antagonism in that relationship, maybe an unease where we don't always share the same conceptual resources or vocabulary, but a way to kind of think together, and becoming a way to do both better science and better critical theory, a way to both contribute to the literatures and environmental humanities broadly construed, as well as the primary biological sciences. So that's kind of a very long-winded response to your question, but it's a great question that you raised. I'm happy to entertain other questions, or also call it and say thanks for a lovely virtual interaction, even if it is a little awkwardly mediated with partial connectivity. Yeah. So we've actually there's been a Whitehead kick recently. So I run the Multispecies Coronavirus Reading Group. So a number of the colleagues and collaborators that I identified in the concluding moments of my talk, we've got veterinary scientists, we've got literary theorists, we've got virologists in our group. So we've been reading some Whitehead together. There's a veterinary scientist, Jakob Zenstag, who came to a talk I gave about a year ago and said, we need to read Whitehead together. So we've been doing that. Stephanie Irv has a great piece about bats that engages with Whitehead that we put on the table. I must admit, I'm relatively new to process philosophy. I'm getting it kind of second and third hand, although we did just delve a little bit into the primary literature about a month ago. But yeah, Whitehead is a very interesting and curious theorist of life. You know, someone who's celebrating pleasure and celebrating, you know, dynamics of the biological world that have largely fallen by the wayside in kind of mainstream biological sciences. So, you know, Isabel Stingers has been a big influence, who's done a book on Whitehead. Whitehead kind of is in the background of Haraway, but I'm relatively new to process philosophy.
Okay. Any more questions? Justin's typing.
Thanks, Amelia. Thanks, Justin. Yeah, hopefully in the post-pandemic world, we can all be in a room together someday.
Okay.
All right. Well, thanks for hosting, Simone.
No worries.
And maybe just one request before the video gets posted. Could we trim off the end just because the Q&A was a little awkward and it might be hard for people to follow?
Of course we can. Yeah.
Okay, great.
So we'll cut the Q&A, and then the chat also gets saved, but it's not often sent out, but just for our records, it's saved. But yeah, most definitely we'll cut the questions off. Yeah, just so that it's your presentation.
Wonderful. Thank you so much for hosting.
No worries. Thank you.
All right. Take care, everyone.
Take care. Bye.
Bye.